Continuing on here. Polymerase. What you just saw was polymerase. Polymerase is the weird enzyme. Okay. No, what that is. Stuck together with helicase. Remember, anything that ends in ace is an enzyme. What you just saw was DNA copying itself. That's what replication is. Polymerase is used to hook complementary nucleotides together. If we left the nucleotides, the A's, T's, G's, and C's alone in a vial, eventually it would build DNA. It would require a little bit of heat, though. All right. Eventually it would, but it would take hundreds of years. Polymerase speeds it up ridiculously at the rate you were looking at. Okay, it's like mass production. It's manufacturing process. Okay, polymerase, poly, forming a polymer because DNA is the ultimate polymer of life. It's used to hook the complementary nucleotides together. It puts the right letters in the right place most of the time. Now, if it gets damaged, it can cause mutations, but usually doesn't work that well. So sometimes toxins, a virus, or ionizing radiation cause the wrong nucleotide to be placed in a sequence of DNA. This is bad, contrary to the wonderful logic of all Marvel movies. Ionizing radiation is not good for you, and mutations do not lead to superpowers. Yes, the overriding majority of mutations, okay, cause harm, with a huge proportion of that death in a simpler organism. So sorry, Spider-Man would be toast. Okay. But it's a cool idea. They cause the wrong nucleotide to be placed in the sequence of DNA. This is called a mutation. Now, if we were to look at this from an evolutionary point of view, from evolutionary biology, mutation is great. It's the only means that biology has come up with ways to produce new genes by mistake. Okay. To me, mutations don't happen enough for that to happen. But okay. We're still studying it. Mistakes like these are rare, but they do occur. Mutations are abnormal changes in an organism's genes. If the mutation doesn't really cause harm, an evolutionist in biology will call it, oh, it's great. It's more variation. When we get to evolution, you'll learn all about that. But to the cellular biologists looking at it, they go, oh boy, that's going to be interesting. And that leaves it at that. The geneticist and biologists will go, that's a mistake. That's just a mistake. That's bad. It's not normal. Okay. Mutations, abnormal changes in an organism's genes occur when there's damage. Okay, when there's damage. Can I scroll?
Probably.
Shush. No, still, you're on cold medicine or something. Just chill. Don't be cute. A segment of DNA that contains the instructions to build one protein, okay, is called a gene. A gene is a segment of DNA that contains the instructions to just build one protein. One gene, one protein. This is a central part of genetics. And on the DNA, there's no, to our eyes, there's no marking point saying this is the beginning of a gene. Okay. But the enzymes look at all the A's, T's, G's, and C's in a long line, and they can recognize it like a street sign. It says, hey, turn here if you want to build insulin. Start reading this gene here. Okay. Proteins can be long, short, but the information is all in the DNA to build them. A segment of DNA that contains the instructions to build one protein is called a gene. Genes control something, and you're going to be hearing this over and over again. They control the amino acid sequence, oh gosh, in a polypeptide chain. You're gonna do this hands-on. So right now it's just gobbledygook scientific speech. Okay. Proteins are parts of you. They're what your body needs to build itself, control itself, be itself physically. Okay. So now we're getting into the tough stuff. How we go from information to building the stuff of you, ligaments, bones, cells. It's all on this information, the A's, the T's, and G's, and C's. The A's, T's, G's, C's, they're going to be read. They're going to be read as instructions. And then a tiny organelle is going to start to build one tiny part at a time, you. Anybody know what that organelle is? It's not the mitochondria. The powerhouse of the cell. It's all you people know. It's called a ribosome. Okay. Don't confuse it with the lysosome that does the exact reverse. And ribosomes. All right. Okay. So, looking at the board here, genes control the amino acid sequence. Okay. Well, okay. Genes are the information. Amino acid sequence. Amino acid sequence. That's the beginning of the building of a part of physically you, a bone, a tendon, a ligament. Okay, that's physically you. Polypeptides are simply young proteins. Maybe you remember this from organic. Polypeptides are simply young proteins that the body needs to be manufactured. It's you. Maybe it's an eyelash. Maybe it's a red blood cell. Maybe it's a neuron. Maybe it's a fingernail. It's got to start with something. These are polymers made up of amino acids. Okay. We're reading information just like you buy a bicycle and a kit. It comes with a manual, all the parts. You read the manual, you build the bike. This is the manual. The DNA is the manual, the bike is you. Polypeptides, young proteins. Does anybody need to see anything above? Okay. Information's complex for early in the morning. We're still writing. Some people are still. Right there, that completes, Roman numeral 1. Okay. Your intro. Roman numeral 2. Let's dive deeper into this thing. Codons, transcription, and translation. Okay. Central dogma of genetics looks like this. The dogma is a set of truths that have been found and then tied together. Why? All of these took a long time to study and learn. Okay. Replication, you just learned yesterday. Replication is all about taking DNA, what we already have, and making more copies of it. We need to do this because cells are making replacements for themselves all the time. You're growing, especially, okay? But even if you're fully grown, we still need to replace cells. And each cell needs to have a complete set of 46 chromosomes in it. That's a lot of data. If you took the information in each cell and you put it character for character, it would be a set of papers, stack of paper 300 feet high in each cell. The letters. Replication is simply making a copy of DNA. Transcription is converting segments of DNA into RNA. We're going to look at that today. specifically a type of RNA called messenger RNA. It's a temporary copy. Translation, which is next week primarily. Translation is the physical building of you, physical building of an organism from the information. It just doesn't go from DNA. It's read and you're built. It's a two-step process. Okay. And the goal over here, it's just protein, but that's you. Okay. Not just physical parts of you, but all of the individual enzymes that manage you. Okay. But anyway, this is the central dogma of all genetics. It took forever to figure it out. So what this shows is that DNA not only serves as a template, a master copy for its own replication, it's also a template for something called messenger RNA. First, you got to write that. You're looking at 50 years of some of the best science our species has turned out, besides blowing itself up. We're really good at that. But a lot of this stuff started just before World War II, looking at, what are these fuzzy looking things inside of a lot of our cells? Well, we're just going to call them chromosomes. Oh, okay. What do they do? We don't know. Well, your body spends a lot of time trying to copy them and sort them and make sure that they go into all the cells. I mean, early biologists were simply going, yeah, whatever they are, they're important. It's got to be something critical. They were right. The first scientists who really worked on DNA, they were nuts. Linus Pauling, and Watson and Crick. Crick was really funny, but Watson is just kind of a prima donna, yeah, jerk. He's kind of he likes to build statues of him. So he knew he was smart, but he's fairly certain he's the smartest person on this planet to this day. So, okay. But they did do major discoveries. Okay. Anyway, replication to DNA. Transcription is the first arrow. mRNA. Transcription, the second arrow. And then protein. Got it? I like to show you the big picture on this stuff first. Okay. You've done replication. Okay, you get, it gets DNA and you make copies of it. Check. Okay. Using the rules, A to T and G to C. Now it's like, okay, so what is this leading to? Transcription and translation. Building you. May I go on? Or not?
Yes.
One person. Anybody else? Everybody's still done? All right. Okay, so the dogma shows that DNA not only serves as a template for its own replication, it's also a template for something called mRNA. The dogma shows that DNA not only serves as a template for its own replication, it also is a template for something called messenger RNA as well. An intermediate step between the information on the DNA and building parts of you. No, I had to resize it. That's better. Transcription, it's move this stupid thing out of the way. Transcription. Transcription is the second step in the dogma. Transcription is making an mRNA molecule that's complementary to DNA. It's like converting. It's like converting Spanish into Portuguese. The languages are similar, but they're not identical. But the message you want to convey in Spanish, you want to convey the exact same message in Portuguese. We have to convert the DNA message into a temporary RNA message. And that's called messenger RNA, mRNA. Transcription is making an mRNA molecule that's complementary to DNA. I know complementary sounds like makes it look better. No. It's its own, no, it doesn't. It's its own version of the message, but in RNA language. The complementary refers back to the complementary base pairing rules. I'm going to pause. This transcription is enough for now.